Very good afternoon, everyone. I welcome all of you in the third day of the FDP program. So today we are basically going to discuss about conformation and functional annotation of gene sequences or protein sequences by applying advanced version of BLAST. Okay, so this is the objectives of today's discussion that the BLAST algorithm as such and the BLAST search strategies. And we will go through some demonstration to see actually how it works and what are the critical points that we will discuss from research point of view. Okay, so first question that comes to our mind that why we need to compare sequences. Okay, so if you have a sequence, if you have identified a sequence from, say, a patient sample, and you are thinking that this sequence might be crucial for this particular disease, then you are actually trying to characterize the gene of your interest, right? So, in this situation, comparing sequences or the section of comparative genomics or comparative proteomics of bioinformatics division plays a crucial role in functional annotation and identification of the target protein or gene sequences. Okay, so here what we do actually, we compare the target sequence or the sequence we are having some query to the all the known sequences so far that is deposited in the databases. Now, there are billions sequences in the databases. The question is that how we can actually compare. The way of comparison is the aligning sequences, and if you have a particular target sequence, you can align in two different ways, either a pairwise comparison or a multiple sequence comparison. The purpose of both is different. Okay, so today we are actually focusing on uh, pairwise sequence alignment. So in pairwise sequence alignment, you know that the dynamic programming algorithms actually creates the better alignment to compare between two sequences. But when we apply dynamic programming algorithm, you know that it is computationally very much extensive. And because of this expensive computational nature, you cannot apply this algo to align all the database sequences. And here in this case, you need to create billions of alignment. So it exceeds your computational power. So in that cases, it is very difficult to apply dynamic programming algorithm. So we actually, in these cases, we go for an alternative algorithm without compromising with the efficiency and sensitivity, of course, and that is the local alignment search tool, that is the BLAST. Okay, so here you can see that manually we cannot compare sequences. So this is the comparison in between two sequences. Okay, so here we can compute position wise comparison, that if I have two sequences of same uh, gene but with altered function, then where are the changes? And we try to understand that are these changes is actually altering the function or not. That is the query we have. Okay, so now you see that this comparison can be done in two ways. One is global alignment, another is local alignment. So global alignment means we are comparing two sequences from one terminus to other terminus. That means if we are aligning protein sequences, that means two sequences will be compared from N terminus to C terminus. If we are comparing DNA sequences, then the comparison will be considered from 5' end to 3' end. Okay. And you know that there are several programs, that is GCG gap program, Needleman-Wunsch programs are there. So now if we consider the local alignment, what are those? So local alignment actually do not compare sequences end to end. That means from N terminus to C terminus or 5 prime to 3 prime. Rather that the local alignment actually it in this local alignment it search for similar sequences. Listen, listen. Yes. Sorry. So actually, the local alignment compares similar regions within the two sequences. Within pair of sequences is basically look for some similar regions. Now you might ask the question, what is the difference and why we are going to do that? What happens if two genes have same function, but the source organism, if they have a long evolutionary history, then due to diversification or species diversification, they accumulate number of mutations across the genes or protein sequences. Now you know that this accumulation of mutation is not random. So in the known data set, what we have observed that this accumulation of mutation is confined to a particular region. And now you see that they have retained their function. হ্যাঁ স্যার অ্যাকচুয়ালি ওরা আজকে থিসিসের একটা ইয়ে হচ্ছে ইন্টারভিউ হচ্ছে ওখানে আছে স্যার মনে হয় ওখানে চলে গেছে আপনি 102 দিয়ে দিন না দেখুন ওখানে কে আছে একটু সুকন্যাকে ফোন করে তারপর ওকে so sorry for the disturbance. Okay, so we repeat again. So, due to this species diversification, the mutation that has occurred within the sequences, they are not distributed randomly. Rather, they have been confined to a particular region. The other regions where there is no mutation or little bit mutation, that means that region is particularly conserved with respect to the other regions. Now, why is this so? So we have actually experiment by mutating these conserved regions, and we have seen that the functionality is lost in case of genes or protein sequences. That means you can say that these regions are very important core regions for the function, maintaining the function of that particular protein. And that's why nature has not allowed any mutation in this region. Now, what happens? Why the now we come to the questions back that why sometimes we prefer local alignment over global alignment. Now, if you see that if you compute overall similarity, now you might get the false result that these two sequences might not be similar if you compare overall similarity. Rather, if you compare local similarity, you might find a local similar region, and that is important for maintaining the function. So in these cases, using local alignment will be much more significant in comparison to global alignment. Now you see that there are several algorithms for creating local alignments. You have Smith-Waterman, you have best fit program in GCG, you have FASTA, you have, see, do not confuse this FASTA algo with the FASTA format. Both are different. Okay. So we have BLAST algorithm. So BLAST algorithm is mostly used in this case instead of Smith and Waterman algorithm. Why? Again, the same point that we discussed in case of Needleman-Wunsch, that the Smith-Waterman algorithm is computationally extensive. So If you apply Needleman-Owens or Smith-Waterman for database searching, you need to create huge number of calculations that is very difficult as per your computational power. So we have to have a different algo which can bypass some of the calculations without losing its efficiency. That was our point. So now this is the pictorial view of global alignment. You can see that in this case, the sequences are compared from N terminus to C terminus or 5 prime to 3 prime. The whole sequence is considered for the similarity calculation. Now, if it is global local alignment, now you can see that if it is our target sequence, so we can map some of the regions, similar regions within the sequence. See if these two regions are similar and both the protein have same function, that means that these similar regions are very, very crucial for maintaining the function, and their source organism must be taxonomically at long pathway. Long distance pathway will be there within these two source organisms. Right. So now there are some key points we'll talk about. That means before going to the advanced version of BLAST, what is our plan to revise once again the basic algorithm that is used for BLAST analysis, and then we will move to the different versions of BLAST. Okay, so that is our basic plan so that we may not miss out the basic things. So we can revise ourselves and we can learn in advance. So that is the objective of today's session. Okay, so in pairwise alignment, you know that to evaluate the score, actually we can apply two scoring metrics. One is called PAM series and one is Blossom series. Now, I am not going in details of what are PAM series, how they are calculated, what are Blossom series, what they are calculated. If you are interested, you can go into details of this PAM and Blossom series. This will give you the idea that when to use which series and which exact version of the series you need to use according to your data set. So that is very, very important, of course. So like here you can see that PAM10 and Blossom 80. I will just tell you that PAM10 means PAM actually percent accepted mutation, or many times we say point accepted mutations also, but it is the basically the percent accepted mutations. That means if it is PAM1, that indicates that this scoring matrix has been designed considering the sequences where 1% mutation has been accumulated. Okay, that means they are very close. Okay, they are from closely related organisms, and obviously these sequences must be evolutionary from evolutionary related organisms. So evolutionary relationship is there. So that is the difference from Blossom. Blossom does not consider evolutionary relationship. That means evolutionary relationship is not considered by Blossom, whereas it considered only functional similarity. Okay, so it does not consider that the taxonomical pathway of the source organism. So if the proteins are functionally similar, they can be considered for creating Blossom scoring matrix. Now, what does we mean by Blossom? Blossom is the block substitution matrix. Block substitution matrix, if you take, so just let me show you. So, say if this is one sequence, two, three, four, five. So say there are five sequences. They are functionally same. They have say function. They are hexokinase. They are hexokinase protein. Okay, from five different organisms, right? So these organisms may or may not be evolution related. So Blossom will ignore that. Now, as they are functionally similar, what you are expecting? You are expecting some conserved regions, right? Say these are the conserved regions. So this is conserved regions. What do we mean by conserved regions? Mutation were not allowed in these regions. Or if there is mutation, some positive replacements might be there, but that is very few in number. But these are the regions, that is non-conserved regions, where the mutation has been accumulated. Okay. Now it means that Blossom will consider these regions only. Blossom will consider these block regions to calculate the scoring matrix. So these regions are termed as block. These regions are termed as, these conserved regions are termed as block. That means that's why the name is so, block substitution matrix. Block substitution matrix. So substitution means if there is a alanine in one sequence, if there is a valine in another sequence, that means A is substituting V or V is substituting A, vice versa. So in this way, we talk about the mutation. Now you can ask me then what is 80? 80 means, see, if these sequences are 80% similar, the overall similarity, not the similarity of this block. Blocks are highly conserved. Okay. If the overall similarity of these sequences are 80%, then the substitution matrix that we will generate by considering the block will be called Blossom 80. What if sequences are 90% similar? Then this substitution matrix will be termed as Blossom 90. Okay, that means Blossom 60 will be considered the 60% similar sequences, but they're functionally similar. So that means there is a series of matrix, and which matrix you will use, that will depend on your dataset and target sequence. That will be varied. So it is not straightforward that you will use any series of the Blossom or any series of PAM. So first you have to decide that the evaluation of the alignment will be carried out by evaluation by PAM or Blossom. If you are interested about, or you are engaged in research with evolution related protein or evolution related organisms, then you must take the appropriate version of PAM, okay, considering the target sequence and your query sequence. Similarly, if you are not interested with evolution relationship, you are interested only with functional similarity, then you have to consider the right version of Blossom. Okay, so that is the logic between the scoring matrix. Okay, so this is the basic definition of BLAST. All of you might be knowing, but I have kept these slides for your quick revisions only. So that was the objective. So you know that BLAST is a very fast algorithm. Okay, it is accurate and it is very easy to use because it is having web access. So sometimes we can use standalone BLAST as per our requirement. So that we will not discuss here. If you are interested, you can contact us. So we can help you in this regard if you need standalone BLAST. So sometimes for research purposes, we may need to use standalone versions because they are the target database and connect your query database has to be say manipulated in that way. Okay. So now the questions that comes to our mind, that why we are going to use BLAST. You can see the applications. It's not restricted to this one, but these are the basic one, that identifying orthologs and paralogs. I hope you are well aware of orthologs and paralogs, and which one is very difficult to identify. If you can tell me in the chat box, I'll be very happy. Okay, so discovering new genes or proteins, discovering variants of genes or proteins, investigating express sequence tags. You know what are express sequence tags? These are very, very important. Okay, these are tag sequences of say 300 to 500 amino acids long generally. Okay, so they can confirm the presence of a particular gene. It's like the tag you get when you are flying. Okay, so for your luggage, you can uniquely identify your luggage by matching some tags. So these are like this. Okay, so and these sequences are expressed because they are part of exons only. So that's why they can be part. They can your cDNA can be means identified by using ESTS. So that's why they are different from STS databases. Okay, and exploring protein structures and functions. So these are some few functions only we have enlisted here. There are so many. Okay, so now there are four important means parameters while for efficient searching of BLAST, first of all, we have to have a query sequence, we have to have a query sequence. So as I said earlier, that what are our query sequences? So you are working with say MDR strain and wild type strain. So wild type strain is not drug resistant, while your MDR strain is drug resistant, right? So now, if you compare the proteome of MDR and say wild type one, then you might get some different bands as compared to normal wild type bacteria, right? So now these bacteria will be very, very crucial for these MDR properties of the bacteria, right? So these bands then can be isolated and they can be sequenced. And these will be your query sequence, right? So this query sequences, that means now you have some amino acid sequences, only you don't have any clue that what the protein is, what might be its function, what are various parameters that exist within this sequence pattern, and what is their functional mechanism, what might be the structure this protein can have. So you do not have any idea regarding that. So that is the start point of your in silico research. Okay, so this is your wet lab research. You will find different protein you will, from the target sample, and then your in silico start research will start. So that is the sequence one, right? Now, in step two, you have to select the appropriate BLAST program. Why I said appropriate? So many times what we do, we have the sequence. Okay, so very easily we can get the BLAST program at the NCBI website. So we go to the NCBI website and we use any one of this version, whichever we have some we are familiar with, right? So that is not the approach. That should not be the approach, right? So what we should do, we should select the appropriate BLAST program, which as per my data set and as per my objectives. Okay, so that's what we are going to discuss today. Okay, so now we have the sequence, query sequence, we have selected the appropriate BLAST program. Now, which database I need to compare? That is even another question. That which database I need to compare. So many times what we do, we just go to the database and we keep the default database, that is the NR. Okay, so that again, that is not the right approach. The NR database definitely you can select. Yeah, I know you have fair idea about NR. NR is actually non-redundant database. So there is no physical existence of NR database. NR actually combines different databases, primary resources, and it actually create a virtual database of non-redundant sequences, and then one by one they start comparing with your target sequences. So when we use NR, actually, if our protein sequence is means not known at all, or similar sequences of my protein is not known, then or primarily for the first time, if you are analyzing your query sequence, you can go for NR, but later on you have to refine your searches. as per the other database availability. Like say, if you need to analyze the structure, or if you need to do the molecular docking or simulations, then you will require the structure of this protein. So there is no means it is wastage of time if you search against NR databases, because you will see number of feeds similar to your query sequences and manually you have to select. Okay, so rather than that, we can direct compare our sequences against the protein PDB data bank. Okay, so there is the options. So if I know that my protein is well characterized, then I can select UniProt database. The UniProt database is a very good resources of information that we are discussing in this FDP program. In the day one, we have discussed in details about UniProt. I hope you remember that part. Okay, so now you see that then after selecting the correct target database, now it comes to choose optional parameters. Optional parameters, it might be optional for students, but for research purposes, it is not optional. It might give you the result. That's why it is saying that it is optional. Okay, but you will not get the your required information, the information you are expecting for your publication purposes, if you keep these parameters optional. So to understand these optional parameters, that which one to select, which one will give you the better accuracy as per your data set, again, it is very required to understand the basics of the algorithm. So you know these are the search options you are familiar with. So I will not spend much time in this division. So this is a P blast or protein blast. Okay, so this is the sequence query box. So in many ways, you can give your input. Like so here in this case, you can see that we can give the accession number on of a particular database, any database. If you give GenBank accession number, if you give EMBL or UniProt accession number, PDB ID, that will also do. Okay, so it can, in the background there are programs that it will take your accession number and it can retrieve the sequence from the databases. Or if you have a new sequence, as we discussed just now, in these cases you can copy paste your sequences here, but make sure that you can convert that sequence into FASTA format. Okay, so this is FASTA format. You know that it is starting with a greater than sign, and this line you can describe your sequences. So this part actually ignored by the software. Okay, so here this line. So that's why your sequence must start from the next line. So from next line, position 1 to 60 per line without any gap. Gap means in two sequences there might not be any gap. So gap is also a character. So that's why it should have only amino acids or base characters. Other than this, it should not be there, and gap should not be there. So if you have a protein of length 61, then it should be 1 to 60, and the next one, that is 61, might be here, must be here. So you cannot say that, okay, I have only one base left, then I can put it here only. So then it will not be a FASTA format. So this is the FASTA format we have briefly revised, right? So so if you do not convert it in FASTA format many times, you will get the result, but the calculations may go wrong. So that's why always it's a good practice to convert your sequence into FASTA format and then you put it here. If you have a sequence that is not derived from the databases, it's your own sequence from your research. Okay, so this is a very, very important area. This is an optional area, but this is very, very important. What it actually says, set subsequences. That means, say, I have a sequence from, say, 1 to 1000. 1 to 1000. I have put here. So, actually, I am interested some part of this sequence. So I am expecting that say from region 250 to 625, there might be some important regions that is functionally important. It may be a domain, there might be some motif or something like that. Okay. So if but whole sequence must be there, then only you can compare. That's why you give it whole sequence here, 1 to 1000, but you write here 250, 250 to 625. Then what the algo will do for creating the alignment only, it will consider the sequence of this region. Means this will be your query sequence now. But as this sequence will be there, you can compare the flanking regions, but this one will show you the actual calculation of alignments. Now this is the region, this is the by default, as I said, this is the NR. So from here, as per your wisdom, you can select the correct database. Like say if you are doing phylogenetic, sorry, ribotyping, then you should go for rRNA databases rather than NR. So if you get NR, then you will get some non-specific sequences, and it might mislead you for creating the phylogenetic tree. The correct sequence you might miss out. Okay, so that's why it should not be taken in that cases. It should be rRNA databases. Okay, for phylogenetic technique. In this way, for particular purposes, you should select very carefully the databases of your interest. So now you can go for the BLAST program. Now, if you know the algo, then you can get the parameters done. So we'll discuss this later on. So this FASTA format, accession number, this is the FASTA format. As we discussed, you know, this is the identification number, okay? And this is the description of the sequences. So this part will not be read by the software. So looking at this sequence, we can understand what the sequence is, okay? What is might this function? That's why you have described this one. But software will read it from methionine, right? M, and it will end it here, the lysine. Okay. So you see there are 1 to 60 sequences per line. This is the 60th sequence, and this might be the 61, and this is sequence position 120. Okay, so this is the sequence. Now you see, if we come to the right version of BLAST program, which version of BLAST program we should select, you can see that there are so many programs. Okay, right? If you talk about nucleotide BLAST, there are so many programs. If you talk about protein BLAST, again, there are so many programs. These are translated BLAST. These are genome BLAST. Okay, that means if you have, if you want to compare against genome, okay, your sequence, you are interested to compare not against the nucleotide or gene sequences of many databases. Instead of that, you want to compare your sequence against a particular genome. It might be human, it might be mouse, it might be rat, or it might be anyone else. Okay, so you can do that. So this is the genome sequence databases, right? Now, what are translated databases and what are the requirement of translated databases? So we will talk about that in details. Okay, this is an important segment. So that is our objectives of today's session also. So these are special databases to align two sequences. You see, so generally what we do, if in the BLAST you have seen that there is space of putting only one sequence, that is your query sequence. The other pairwise alignment requires two sequences, right? So the next sequence it will retrieve from the database. But sometimes it may happen that you have got two protein. Okay, might be say one person is having, say, hemophilia, and another is the healthy person. So you have got the respective protein and you want to compare this one. So in BLAST, you will say that we can't do BLAST. If we do BLAST, it will compare against databases. So I want to compare these two sequences only, right? So in that cases, you can use BL2 sequence, this one. Here you will see that there are options for putting two sequences as input. Okay, so that is the difference of this one. Now there are some special blasts like immunoglobulin blast or IG blast. Sorry. So this is immunoglobulin or IG blast, right? So there are, now you can understand that there are so many blast programs. These are actually modification of basic blast program. Okay, so they are specialized for analyzing the specific data set. So as per your data set, you have to choose any one of them. It's not just like I can use any one of the version. Okay, now see, I was talking about the translated blast here. So blast N. So most of you basically use either blast N or blast P. Okay, so what they are doing, so first one is the nucleotide blast. That means here your query sequence is a nucleotide or gene sequence. Okay, gene sequence of particular gene. It's not the genome, please. It's the gene sequence. You know the difference between gene and genome. Now, and what is your target database? Target database again, it's a gene database. So you might ask the question, sir, it is obvious that if I have the query sequence gene, then my target genome, target database must be the gene database, nucleotide database. It's not true. It's not true. Just wait while we will discuss this part. Okay. So in these cases, you will use BLASTN perfectly. That I have a nucleotide sequence and I want to compare against all the other similar genes. Right. So this I need to compare. Now, take similarly, if I have a protein sequence, okay, if I have a protein sequence and I want to compare it against the protein databases, then I will use BLAST P. So what is my interest? My interest is that I have a protein sequence like we discussed earlier that from the disease sample you can get a protein sequence and you want to know that is the same protein is known or if similar protein of your query protein is available in the databases. If you get those proteins, you can compare and you can predict the function and functional insights of your target protein and you can carry on your research in this direction. Right. So that is the objectives of BLAST P. Now the point comes to translated BLAST. Okay. You will see that there are three versions of translated BLAST. One is T BLAST N, BLAST X, and T BLAST X. So both these three are translated BLAST. So when actually we use translated BLAST? So say if you have a protein, you have carried out BLAST N or BLAST P, whichever is suitable for you. You have done that, but you have not found any input, any output. You have got the message that no output was found, no output was found. Then actually we think that it might not be known or I cannot analyze my sequence by using BLASTP. This is the point where we go wrong. Okay, so this is the point you will use BLAST, translated BLAST. So mainly translated BLAST is used for research purpose. Okay, translated BLAST research purposes, we use translated BLAS. Now let's just see that how actually they work. Then we will understand that which translated version I need to use when and how actually they are functioning. Now you see that if we have BLAS 10, you know that our input is DNA and target database is DNA. We call it 1:1 cardinality. That means one nucleotide sequence will be aligned with another nucleotide sequence from the database. Similarly, the same with BLAST P. Okay, so you have input sequence protein and input sequence protein databases. So here also it is 1:1 cardinality. So this is 1:1 cardinality, and this one also having 1:1 cardinality. That means 1:1 comparison. But here you see the differences are there. See, this is BLAST X. So if it is BLAST X, then your input sequence is DNA, right? Your input sequence is DNA. And you see your target database is protein databases. Now you might ask the question that how we can compare or align a DNA with a protein because they are biochemically different. So we cannot compare. You are absolutely right. We cannot compare. But what happens to increase the sensitivity of the BLAST program because you are using these versions only for research purposes, right? So when you are using these versions, when you have not got any output from these two versions, or enough sequences are not there, you have got very little information, then you are moving in this version. That means here what will happen that your DNA sequence or gene sequence, nucleotide sequence will be computationally translated. If you computationally translate your DNA, you know that we can translate it into 6 reading frame from the plus strand and minus strand. For the plus strand, it's the 3, and minus strand, it's the 3. So 6, it will be converted into 6 protein sequences. That means instead of getting one protein in this case, you are using input of 6 protein sequences, right? 6 protein sequences. So now this 6 protein sequence will be considered your input and the database protein sequences then will be mapped for similarity. Okay, so that means a huge increase in sensitivity of the program. Huge increase in sensitivity. Now you have to keep in mind that when sensitivity increases, then specificity will decrease. What do we mean by that? If you increase the sensitivity, then you can expect that lots of false positive might be there. So you need to be careful in these cases. You will get information, definitely, but in this information, there will be some errors or false positives. So manually, you have to go through that part and you have to carefully read it out. Okay. Now, if we talk about T-blastine, okay, in T-blastine, it's just opposite. It's just opposite, right? Okay. So here you say that here your input sequence is protein, okay, and the target DNA, target database is DNA database. That means the DNA sequences now it is translated into 6 reading frame. The whole database, see, there are 1 billion sequences. Now, these 1 billion sequences will be translated into protein sequences into 6 reading frame. That's why they will take some time. It's not as fast as BLAST N and BLAST P. It might take some more time, and the time will depend on the your system and your net connectivity. Okay, so it will vary from one individual to other. But overall, the calculation times will take more as compared to blast N or blast P. Now you see that it's a huge task, right? To blast in the DNA, he blast in the DNA whole database has been sequenced into 6 reading frame, right? So now the last one. So in these two cases, we are saying that it is 6 to 6 cardinality. Cardinality where 6 is to 6. 6 is to 6 cardinality. Now you understand that why it is called 6 is to 6 cardinality. Now if I talk about T blast X, okay, it is T blast X. Now here in this case, you see it's just like blast N, it's just like blast N, but it is actually not. Here also input sequence is DNA, target database is DNA. But here what happens? The input DNA will be translated into 6 reading frame. That means you are having actually the protein sequences as input, and you have chosen the target database is DNA database. Again, similarly, like blast N, the DNA database sequences are also translated in 6 reading frame. That means 6 into 6, the cardinality is 36. The cardinality is 36. So many times in NET or GATE exam, students finds this sort of questions that what does the cardinality mean and what is the cardinality value for T plus X or T plus 10 or plus X like that. Okay, so these are the cardinality all about. Now you can see that huge sensitivity will be here in this case. And at the same time, you have to keep in mind that lots of false positive will be there. Okay, so nature knows that where is the position of start codon. So it actually considers the 1 reading frame only. Okay, so if we are not sure about this one, then we computationally predict the 6 reading frame, and out of the 6, one will be translated by nature. So it is the 6 is to 1 is the speciality. Okay, so this is the concept of 6 reading frame. You know, I am not going through this one. So here you can see that how the coding is basically changed. If you miss one base only, so if you miss one base, then you can see the coding frame will be different, right? In this way, for one strand, there can be 3 different coding frame. For the other strand, you will have the other 3 different coding frame. That's why it's called 6 reading frame. So these are the data sets. So almost there are 32 databases in the database. So I hope you should have a fair idea about all these databases, what their meaning, and what type of database will have what type of information, and when to select which database. You should have a clear idea about that. Now see, if you are interested about conserved domain, so in the last session, we discussed about conserved domains, how efficiently we can get the domains for a particular protein from the UniProt. But if you need to search domain, then you have to apply some means conserved domain search programs. Like in the yesterday session, we discussed about how we can find out a domain by applying cat and scop, right? So these are the some other advanced options. So you can put or you can apply your input sequences by putting Antris query. Antris actually it's the name of the search engine that GenBank applies. Okay, so you are writing some query, you are retrieving sequences. So this is a application. This application is termed as Antris query system. So this is our Antris query system, right? So now you see that you can choose some filter like low complexity regions. So these are nothing but the regions of repeat regions. So in especially in DNA sequences, might be some repeat regions can exist, and because of these repeat regions, you can get higher score, but they are not informative regions because these regions might not contain any genes or functional units. So that's why we need to remove these regions. Then we will get better accuracy. Okay, expect value 10. What do you mean by that? Expect value 10? By default, this is 10. Okay, so you can change this one. So if I write expect value 5, I can write that. So how it actually means alter the algo. So in the means background, the cutoff values, the threshold values will be altered. The expect value means that how many sequences I am expecting by chance. By chance means they are mathematically similar or statistically similar, but biologically they are not similar. That means here expect value 10 means if I get 100 sequences in this search, out of 100 sequences, 10 might be by chance. If I write similarly 5, that means out of 100, 5 sequences are there by chance. So that is the information about expect values. Okay, so if we see the calculations, shortly we will discuss the calculations before the demonstrations. Then you will understand these values. So how they can means alter the calculations, these putting these values very easily in the background. So that's why many of us we like to use BLAST because it's very user friendly. See, we are not writing any code or we are not manually say adjusting the algo values or weightage of those algorithms. Rather, just we are taking giving some human understandable parameters, and accordingly the algo is modified in the background. So that's the beauty of this program, and that's why most of the people, almost every day, the scientists, the especially biological scientists, they use BLAST program. So that's why we thought that the discussion regarding this tools will be an important aspect for the FDP. So now the next one is the size of word. So by default, you can see 3. In case of protein, it is 3. In case of DNA, it will be more than 3. Might be it is 11. Okay. So what size means? So it's a k-tuple based method BLAST. K-tuple means it actually consider the query sequence as a sentence actually. It does not understand what is A, what is B, what is C. It does not understand that. Okay. It does not understand that. So rather, actually, it considers like a sentence and it's breakdown into number of words. So we will discuss that shortly. Okay. So now these words are actually compared against databases. So this technique, this type of algo they are using to bypass many calculations that actually considered by Smith-Waterman, and that's why they are so efficient and specific in searching databases. So in case of searching databases, people do not use Smith-Waterman. They can use BLAST only because of this one. So now the length of this word will, if you increase the length, that means lesser number of words will be there. So if it is 2, then if these many words are possible, then if I make it 4, that means I will have lesser number of words. The word will be 50% lesser. So if you have 50% lesser, you are going to have more specificity. You are having lesser sensitivity. Okay, so in this way, the word size will increase or decrease sensitivity and specificity accordingly. Okay. Now, of course, it's scoring matrix. As I said that choice of scoring matrix is a critical point. Okay, because depending on this, all the calculations will depend. So in your manuscript, you have to explain all these parameters. Otherwise, reviewers will ask many questions from this part. Okay. So this is very, very important. And if you write this part explicitly, if you write a nice paragraph of the background strategy of your application of BLAST program, then I hope the reviewers will be more happy regarding that knowingly you have done all these things. Okay. So there are some advanced versions like say PSSM. So you know PSSM is nothing but the position specific iterative matrix. Okay. Position specific scoring matrix. What actually, why we use PSSM? PSSM actually used instead of our query sequence. So, what actually happens if we use any just last, like yesterday I was talking about this point that if we use a particular sequence you consider that say this is the albumin protein. So I'm using a sequence of albumin and I'm expecting that from the database I will get all the albumin sequences. So now the sequence I am using that is not the representative sequence of albumin. That means if I am getting similar sequences of my query sequence, I am getting bias to this sequence. Okay. So the things I will not repeat today because we discussed in details in the last day, right? So here, if we use PA system instead of our query sequence, we can remove this biasness and we will more efficiently, we will be able to retrieve some more positive values. Okay, so that is very, very advanced one. So this is all about this one. So now you can choose this one optional parameters as I said, as we discussed. So in the output, you might see some these are the low complexity regions. That means the repeat regions. So these are the filtering part. So outputs already you have seen in your blast you have getting this sort of output, so I just wanted to show you this part that whatever you are doing so far we are going similarly with you, we are not discussing anything new. So we are here only to discuss about some tricks so that you can use this efficiently. Okay, so outputs you can see in alignments view, descriptions view, okay, and you can go through the alignments, okay. So these are the options of the algorithms. So after the algorithms, you can change this part, okay. After the, you see, this is for SciBlast, okay. So you can use this one and you can increase the accuracy of your tools. So these are the program. The output will show you that which program you have used, the reference for this program. That means if you are using this for your research program, so you must use this citation. Okay, this is the citation for BLAST, and this is your query sequence, the length of query sequence. You see, then database how many sequences they have compared, okay, and how many letters. As I said you, that BLAST does not understand protein sequence or DNA sequence. They consider it as sentence. They break down into words and letters, okay. So these are the letters they have searched, these many sequences they have searched. They have compared these many alignments, okay. So now the taxonomy reports is also very, very important. So these are taxonomical position of your output. It's shown in this way, right? So these are high scoring, high scores and lower e values. You see, so you will see that lower the e value, better the output. You see that it has means listed the outputs in the uh, increasing order of say uh, e values, right? So now this is the alignment option. You can see these are the alignment option. Here also you can view the sequences in multiple alignment options also, along with pairwise alignment. So these are the multiple sequence alignments, and these are some basic points. So this was uh, given why the main approach to sequence alignments, And Smith Waterman we have already discussed, and this was the point that was quoted by Asul. Okay, and he is the scientist who devised this algorithm. Okay. So now the algo, as I said, that we'll briefly discuss this algo. We'll not go in details, but briefly we will discuss these algorithms and then we'll go for the demonstration. Now you see that if we look at this BLAST algorithm, you see that this sequences, this is a query sequence. Okay, as I said earlier that it's a k-tuple based method. That means k actually it's called k-tuple based method. BLAST is a k-tuple. Tuple means actually word. Tuple means word. Okay, that means it's broken down into words. That's why it's called k-tuple. Then what is k? K is the value or means length of the word. That means k value can be 1, 2, 3 like this. 1, 2, 3. That means if value of k is 3, that means 3 amino acids will be broken out as words. 3, 3 in overlapping manner. It's just opposite your genetic code. Okay, genetic code, you read this in non-overlapping manner, right? And this sentence, that means amino acid sequence will be broken down into words, and these words are in overlapping manner. That means, say, if it is A, T, T, C, G, that means A, T, T will be the first word, then T, T, C will be the second word, T, C, G will be the third word accordingly, right? So in this way, you will first break into words. Now, these are the words possible. L is the length of the sequence. W is the length of the word plus 1. So that many words actually you will get if you break down a sentence. Now, in next step, what happens? These query words are used to increase the sensitivity of the program. How? Say, assume that you have 20 amino acids, right? So if you consider the length of the word is 3, that means 3 to the power 20. That means 8,000 words is possible total, right? Total number of word can be 8,000. Now, within these 8,000, this actually we call database of word. Database of word. That means these many words are actually possible. So, as I said earlier, that it's a beautiful program of statistical program. You see how beautifully the statistics can retrieve the biological significance. So here you see that these are the database of words. That means using the amino acids, all possible words are here. That is 8,000. Now these words are also here. So you will get the exact same word here. If it is exactly same word here, then if you score them by allowing the blossom or Pam, so for the position wise matching, you will get some score, you get the summation of that. Three letters are there. Say for one letter you have got score 5, other 6, and the next one again 4. Then what is the total score? 5, 6, 11, and 4, 15. That means 15 can be the maximum score for this particular word. Then it can be 14, then it can be 13. So you can put a cutoff value. That is above 20% decrease. If it is in decreasing manner, say 20% cutoff. That words will be considered as the similar words for this word 1. The word 1. So these are the 10 words. Say if you are getting 10 words in this way. That means for each of the words you got in step 1, in step 2, you will increase the number of words by comparing it against the database of words. So there actually this plays a key role in retrieving the orthologous sequences, because you know that orthologous sequences are functionally similar sequences, but they might exist in distantly related organisms. Okay, they accumulate lots of mutations. So that's why this is the key point, that the BLAST is efficient to retrieve these sequences. Okay, now you see that in next step what they do, this step they are a little bit tough. So the words now will be mapped on the database sequences. Wherever it will map exactly, only that positions will be called as seed alignment. Seed alignment. Okay, it is called as seed alignment. Now, this seed alignment can be extended in both direction. Okay, these are the seed alignment. These are the seed alignment. Now you might think that it is matching only here. So all the other words that will match. So if the two sequences are similar, you will see many number of words matching. If not, that means only one or two by chance matching. So these will be rejected. It will not be considered for further calculations. That's the crucial point, how BLAST bypasses most of the calculations. And that's why within a few minutes, it can create billions of alignments, and it can report the similar sequences of your query sequence. Okay, so this is the right point where it actually bypasses the unnecessary calculations. Now you see that if sequences are similar, you will get number of seed alignment on it. Okay, and next, you can extend the word by comparing this in the both direction till a threshold value. Means if the after matching it, if the getting unmatched and sequence is going down, then you can put a threshold value so that e-value word length it will increase this or decrease this threshold values, and they will modify the program. Just you have we have discussed earlier, right? So in this way, you can get the MSPs. Now, if two MSPs are very close, then you can align them all together and you can call them HSPs or high scoring segment pair, which are actually the similar regions. Okay, aligned regions. So now, what are the statistics? This is very, very important for us to understand, and definitely we will discuss a few minutes here. So, in the BLAST output, you have seen identity, right? You have seen identities, okay? Identities. You have seen similarity. You have seen e-value, okay? So, these points are very, very important. So, identity means number of nucleotides or amino acids matched exactly in between your query sequence and the database sequence. In this, your query sequence or the database sequence. So these are the two sequences. So in between these two sequences, so what are the matching? What are the matching? So that is the identity. If in this position A and in this sequence also A, that means it is identical. Okay, if it is A and there is another other than A, but that from the same chemical biochemical group, then it will computed as similarity. So important for protein alignments. That's why. And E value, as we discussed earlier, that it's the expect value, that how many sequences you are described number of hits can expect to see by chance. Okay, you see, this is by chance. Okay. Now, again, you will see there will be score values, the maximum score, you will get the highest alignment score or HSP just we discussed, and total score, the sum of alignment scores of total segments from the same subject sequences, right? And here is the point I was searching for. This is the query coverage. Okay. So many times you have seen that the query coverage and identity, these two parameters, along with other parameters, are the most crucial parameters that can alter your decision or that can influence your decision. Okay, so many times if you see that your sequence is having query cover is 100% and identity 100%, that indicates the same sequence. That means your sequence is not novel. Okay, the sequence you have retrieved from your lab already, some people have worked on it, but your objective and their objective might be different. Definitely, you can go through the database, you can go through the files, and you can check that what they have done, and that might be different. If you have same objective, it's your luck that you have checked that, and you will not repeat your work. Okay, so unknowingly, if you are repeating your work, then after a year or two, your manuscript would get rejected. Okay, and then you would come to know that you have done some repetitive work. So that's why it's good for you. But if your objective is different, then obviously the protein or gene might not be novel, but you can work on that. Okay, but if it is query coverage is 100%, query coverage is very, very crucial. Query coverage is 100% and identity is 100%, then it is the same protein. Otherwise, it will be very close. So many times it may not be the close or similar sequence may not be 100% query coverage, but it should be as good as more than 90%. Okay? Otherwise, you should not consider the sequences as similar sequences. Okay, so now this is the calculation for e-value. As I said, that lower the e-value, better the significance. Now we can see that why I am saying this. See, actually this indicates that division of y, right? If you let me explain, so this is the division, na? Minus lambda s. So lambda is a constant value. This kappa is a constant value. M and n. M is the length of the sequence and n is the length of the letter. Earlier we discussed that it shows you the report that how many letters it has considered, what is your sequence. So the value is derived from these two. Okay, now E is the again a constant value and it is divided by that. And S, actually S is the score. So you have got the maximum score also. It is reported. So if more longer regions, the S will be higher. Okay, score will be higher because the sequence is more longer. If S is higher, then you are dividing the by S while calculating the E value, so value will be lower. Okay, so it's like that. That if a student is doing better in one exam, like one internal exam, so we cannot say that this is a good student. But continuously, if it is doing better, if she or she is doing better in internal one, internal two, internal three, in their exam, then obviously he or she is a good student. So that's why if similarity means when you are aligning these sequences in both direction and they are getting similarity, right? They are getting similarity. So they are a biologically similar region. Then they are considered as biologically similar region. Okay, so more longer the regions, they are more biologically similar. So that's why e-value goes down. So that's why we say that if lower the e-value, better the significance. So similarly, this is the bit score. Just I told you that how it is calculated, and this is the probability value. That what is the probability that they will be there by chance. So now, see, this is the basic brief revision, you can say, your algo. So I know you know all these things, but we quickly revised. So we did not go in details of this algo, but we enough we have revised. Now we are in a position to take some research related problem and we can go on for hands-on demonstration, right? So say here many times we get this type of problem. So from the problems that is asked by the research scholars for their research projects, so I have taken one question from there to show you that how actually we should plan. Okay, so here you see that the question is that illustrate the similarity. Illustrate the similarity and differences between the GAPDH gene in human and chimpanzee. I repeat once again, illustrate the similarity and differences between the GAPDH gene in human and chimpanzee. Okay, then what they have asked further? They have asked that mention the genomic coordinates in human and chimpanzee genomes. So for the genes, what is the position? Basically, it's asking that what is the position of the gene? What is the position of this gene in human as well as in chimpanzee? The position might not be same because these two organisms are different. They have different sets of chromosomes, right? They are very close. Chimpanzee and human are very close. We have only 0.5% difference. Okay, we have only 0.5% difference. Because of this 0.5% difference only, we visit the zoo to see them, right? So think about if it was vice versa, then what would happen, right? So now we say that the position, the GAPDA's position in human and in chimpanzee. How we can find out that? And then the second point is asking that quantitate the extent of similarity. Quantitate the extent of similarity. So what is the similarity in between these two genes? Okay. What is the percent identity? What is the similarity? And what are the other features? How we can explain that? So that is the point. So let's end it here and let's go to the web for this point. Okay. So here, see, I will go to for the genome sequence. I will go to UCSC genome browser. UCSC, see, this is UCSC genome browser. So you can type the web address like this one genome.ucsc.edu, or you can directly write into the Google and you can go over here. That's not an issue. Okay. So this is the UCSC genome browser. Okay. So from here, this is the home page. From here, you can check the genome browser from here. Okay. So this is the genome browser and there are some other resources you can see. So directly we can go here for genome. See, this is the genome browser gateway. Now you see that popular species are there. So it is taking some time. Okay, just wait. Yes. So you see that by default, human assembly is there. So you know that we have huge number of genomic sequences. So sequencing of human genome earlier it took 10 years around and around $1,000, $10,000 for this sequencing. Now you can sequence a sequence in a day, and the cost might be say $100, less than that, right? You can get the genomic sequences. So say if it is if we need other genomic sequences here, see we can change this one. See if I need this one, marmoset. So I can check this one. See, marmoset assembly has been selected. So in this way, I can select the species of my interest. Okay, from here for this genome problem. Okay, so there is a huge list. So all the genomic sequences available, there is a list here. Okay, so there is a list for the whole genome sequences. So now today we are interested in human, then we can compare against chimpanzee, right? Okay, so we have to go to human assembly. So now we have checked again the human assembly. Okay, so this is the human assembly. Now, see, we do not know the position. We can search through positions. As we do not know the positions, we can write the name of the gene. So we are interested in gap DH gene, right? That was the question. Gap GA. So now if you see gap Gs, you can see that there are so many variants. So we will select this one. So now we will hit go. Just wait. The browser is going to be downloaded. See, here you go. Okay. Now you see that you have got the whole chromosomal sequences of human. Okay. Now you see that your genome, your gap DH gene is on chromosome number 12. Okay, it is reported in chromosome number 12. And the position, start position of the base is see, 6 lakhs, this one. Okay, 517, 534, 6. So these are the starting position and this is the end position. And what is the length of the gene? That is the 3855 base pair. Okay, this is the length of this gene. Okay, now you can mark this one for your reference in your manuscript. You can cite this position. It is in the chromosome number 12, and this is the start position, and this is the end position, and this is the length of the sequences, right? Okay, so this is the genome map. It is showing you the different regions, right? And so many amine alleles, okay, left secreted amine genes, and so many things. So if you have an idea about the different databases, so you can really enjoy this one. Okay, so see, if you know all these things, then only you can customize your research. So what you should do, you should take some time. It will test your patience, I know, but these are self-explanatory. Okay, so you can go to the next wave and you can see what are the meaning of each of these regions. Okay, so see, there are variations. If you want to see DBSNPs are there. Okay, so you can mark the variations here. So DGV structural variations are there. So many, so many. So it's a really a beautiful platform. Okay, it's a really a beautiful platform. But the thing is that you have to explore all these parts. Okay, see the spliced ESTS. Okay, so you have to explore all these things. You have to know all these things, and then only you can do a lot of research by using these tools. Okay, so now what now we are not going in that part because that is not our today's objective. So our objective is to get the GP gap DH gene. So from where we can get the sequence? Now we need the sequence. Okay, so for the blast sequences, because we need to compare against chimpanzee, so you hit the view option. Okay, so if you hit the view option, you will see that there is an option called DNA sequence. So now if you check that one, see you have got a window, right? So to retrieve your gene sequences. Now you see it is marking the positions. So we have searched this position, that's why it has marked that positions. Now say if you are interested to get the PCR of this gene, so you want this gene intact. So now you can go for 1 kb or 500 base pairs in the upstream and downstream. So you can write there. Okay, because you are expecting that you will get some cut site in that upstream and downstream regions, and you can cut out this whole gene if this is your interest. So now we are not checking that because we are not interested in this one. So you can manipulate in this way. So now what you can see that we can mask the repeat regions or lowercase regions. Okay, so we are not getting, we are not interested in reverse complement. So you might be careful about reverse complement. So next day, tomorrow's session, I will show you that how the negative strands can play a major role and it can bother you while creating phylogenetic tree. So tomorrow's session, I will show you. So now, what is our objective? To get the DNA. So we will get the DNA. Now you see that we have got the DNA. So this is the DNA, that is the range from chromosome 12. This is the base coordinate position. This is the start position, and this is the end position in human. Now we have to compare this position against the chimpanzee. That shortly we are going to do that. Okay, so this is the plus strand, right? The strand is plus and repeat masking. Okay. Amra, we have masked this region to lower cases. If there is any lower case regions, that means that is the repeat sequences. Okay, so if there is no regions in lower case, that means we do not have any repeat regions. So this is the sequence. So you can copy this sequence and you can in you can so you can save it into raster format. Okay, so this is your first sequence of GAPDH gene. This is the position. One answer you have got. Now the next answer is that uh, how to comparing with the chimpanzee gene, right? So here you have to use the blast search. You have to use the blast search. So here we will select which versions of the blast. So now you think that most of the people time what people do, they go for nucleotide blast as we have the means nucleotide sequence and we are expecting similar nucleotide sequence. So obviously they are going for end blast. So this is the point they are making mistakes. Okay. Here, see, if you select this one, you will get lots of high similar sequences which are not actually you are interested in. You are interested in only in chimpanzee sequences, right? So you just directly you go to set your blast in such a way that it can align not against any databases other than chimpanzee. So I will select chimpanzee. So when I am writing chimpanzee, you can see that these are the chimpanzee. Okay, and this is the taxonomy ID. Okay, chimpanzee, you see the same taxonomy ID. So this is the taxonomy ID for chimpanzee. So now I will select against chimpanzee. And now you see that I know all of you have used BLAST. Now you see the difference here. Okay, you see the difference here. So if you are not doing that, then you could get NR databases here and list of other databases, right? So many of us is familiar on that, right? So that is the case. So this now you see that now we have the sequences. So this is the sequences. So from to this part, I am not going to discuss because this is not our objective today. Those are the basic part of BLAST. So today we are interested only that how efficiently we can manipulate. In just one example, actually, I am showing. Okay. So now this is the sequence. So are there algorithmic parameters? You can see that word size is by default 28. If it is the nucleotide, it is 28. So if we want to make the program more sensitive, we can go for higher number of words. If we go more specific, so we can go for lower number of sequences. So we can go for 16 words. and accordingly. So maximum target sequences we are expecting 50 only. So accordingly, the threshold values will be changed, right? The gap cost, that is the ideal gap cost, we are keeping it. Okay, so that is the mass mismatch scores. So marks filter out low complexity regions. Okay, repeat regions. So all these things will be taken care of. So now you can hit the blast. Now it will take some time. It is creating alignment against your genome only. That's why you see that it's so fast. Okay. Now you see the output. Okay. So now you see that there are four important parameters. So this is your job title. Okay. This is your program. It has used blast n. So we have used blast n. But we have customized the database. We have discussed how we have customized these databases. So our molecular type is this, and this is the query length. Okay, now our output is there in four sections: the descriptions, graphical summary, alignments. Okay, and there was some error in loading alignment, but it will be done. No, don't worry. So this is the taxonomy. Okay, so we have aligned against only one organism, so that is the only one taxonomical positions is there. So just we'll go for this one. Here you can see that maximum score, total score, query coverage, okay, e-value, and percent identity is there, and this is the accession length. That means, see, the first one is the most closest one. The query coverage is 100%, right? But identity, you see, identity is not 100%. So you see, the first of all, you have got the information that in human and in chimpanzee, although we are the closest organism, okay, the GAPDH gene is not identical in human and chimpanzee. They have a lot of difference. See, overall difference is 0.5%, as I told you. But in GAPDH, you can see close to 2% difference. It's a huge, right? So if it was 100%, then we could say that in human and in chimpanzee, both the sequence are identical. But see, here is the difference. Now, the next one, you can see that there are query coverage is going down and down. It's 34%, so this is not reliable. It might be hit due to some repeat regions of the sequences. That's why you have got the hit. So you have only one choice in this case, so this one. So it's not true that always the first one might be your choice. So as per the calculation of these values, by considering these values, okay, you have to select the right one. So in this cases, this is the first one. So what we will do, we can get this one. Okay. So this is the, we have got the link for the GenBank file, and you see that this is the chimpanzee GAPDH gene. So this is the chimpanzee GAPDH gene. So we can get this sequence, and we can store this one. And now here you can see that the question was this, that uh, this one, the question was that quantitate the extent of similarity. Okay, now we can see that the query coverage is 100%, the similarity is close to 98.3%. Now you can see that where are the differences. So you can go for the alignment. So there are some error in alignment. Just wait. It's loading the alignment. It's facing some error. Server might give you some error sometimes. See, now they have come. Okay, now, so the first one we are interested in, right? So now again, you go to description. Just get the alignment of the first one. Okay, see, this is the first one. So now you see the position. Okay, so we have to mark the position also. So this is your query sequence, right? This is your query sequence 1 to 60. Okay, now this is your chimpanzee sequence. Subject actually indicates the database sequence or the sequence you have compared against, right? So this is now you see the position. You see the position is human and in chimpanzee is totally different, right? So gene is in different position. Gene is not in the same position also, right? You see, this is the start point of the position, right? 7900. This is the start point. And what is the end point of the gene? See, this is the end point of the gene. So you can compare against human and you can report this one. Okay, in your manuscript, you can report this one, right? Now you have to see that what are the other similarity and dissimilarity. So this is the scored values. So this is the expect value 0. Okay, that means it's highly accurate, it's biologically significant. Okay, identities. Now you see that out of 3873, 3807 are identical residues. Okay, that is 98%. And there are 21 gaps. That means these many mutations are there. So this is a strand. What type of strand? Plus strand. Now, if you go back to the alignment, you see that these positions are identical, right? C and C. Now, here you can see that there is a dissimilarity. This is not gap. This is dissimilarity. So in human, in this position, there is C, and in chimpanzee, there is T. Now, by saying that this one, you cannot say that the protein sequence also might be different. You have to see that what the code they are giving. So, if the code is similar, they can code for similar amino acids. So that you have to keep in mind. So in this way, you have to analyze. Here in this position also, you see the G and A, right? So there is differences. So now you can see these are the low mass regions. Okay, so these are the complex regions, low complexity regions. So the gaps are here. So these are the important regions you can see. So either in human, this sequence has been deleted, or in chimpanzee, this sequence has been inserted. We do not know. That we cannot tell. But it has happened or either it has been deleted from human gene or it has been added in the chimpanzee during the evaluation. So this is again a very, very crucial information. So you can generate this algo um, this alignment can be applied into different softwares, and for publication quality pictures can be generated by using different tools. Okay, so these are the total alignments, and this is all. Okay, so now you can report your sequences and its other statistics that has been asked. Okay, so many times your reviewer might ask some queries and you can solve in this way. Okay, so today what we have learned. So this is our today's takeaways in day 3. We are remaining with 2 more days. So we have got blast overview and types. Okay, we have discussed different types of blast. Okay, then advanced blast techniques, and then we have practical approaches for this one. Okay, so these are today's takeaway. Thank you for joining. If you have any queries, I am ready to answer. If you have any queries, I am ready to answer, please.